Unit 2 Cellular Organization 1. Membrane. The cells make use of many different types of membrane. All cells have a cytoplasmic membrane or plasma membrane that functions to separate the cytoplasm from the surroundings. In the early days of biochemistry, the plasma membrane was not accorded many functions other than this one of partition. We now know that the plasma membrane is also responsible for 1. Exclusion of certain toxic ions and molecules from the cell. 2. Accumulation of cell nutrients. 3. Energy transduction. 4. Cell locomotion. 5. Reproduction. 6. Signal transduction processes. 7. Interaction with molecules or other cells in the vicinity. 1.1 Membrane structure. The biological membrane are uniquely organized array of lipids and protein. The lipids found in the biological system are often amphipathic, signifying that they possess both polar and non-polar groups. The hydrophobic nature of lipid molecules allows membranes to act as effective barrier to polar molecules. The polar moieties of amphipathic lipids typically lies at the surface of the membranes where they interact with water. The proteins interacts with the lipids of membranes in a varieties of way. Some proteins associated with the membranes via electrostatic interactions with the polar groups on the membrane surface, whereas other proteins are embedded to the various extent in the hydrophobic core of the membrane. Other proteins are anchored to membrane via covalently bound lipid molecules that are associated strongly with the hydrophobic membrane core. Spontaneously formed lipid structure monolayers and micelles. The amphipathic lipids spontaneously forms a variety of structures when added to aqueous solution. All these structures form in ways that minimize contact between the hydrophobic lipid chains and the aqueous environment. For example, when small amount of fatty acids are added to an aqueous solution, a monolayer is formed at the air-water interface with the polar head groups in contact with the water surface and hydrophobic tails in contact with the air. Few lipid molecules are found as monomers in solution. Further, addition of fatty acids finally results in the formation of a micellus bilayer. A lipid bilayer. The lipid bilayer consists of a continuous arrangement of monolayer. The phospholipid prefers to form bilayer structures in aqueous solution because their pairs of fatty acyl chain do not pack well in the internal of micelle. The phospholipid bilayer from the rapid and spontaneously when phospholipids are added to the water and they are stable structure in the aqueous solution as opposed to micelles, which are small self-limiting structures of a few hundred molecules. The bilayers may form spontaneously over large areas 108 nanometer square or more. Because exposure of the boundaries of the bilayer to the solvent is highly unfavorable, extensive bilayer normally wrap around themselves and form a closed vesicles. The nature and honesty of these vesicle structures are very much dependent on the lipid composition. The phospholipids can form lipid composition. The phospholipids can form either unilaminar vesicles with a single lipid layer known as liposomes or multilaminar vesicles were discovered by Sir Alex Bagham and are sometimes referred to as bagosomes in his honor. Liposomes are highly stable structure that can be subjected to manipulation such as gel filtration, chromatography, and dialysis. With such method, it is possible to prepare liposomes having different inside and outside solutions composition. The liposomes can be used as a drug and enzyme delivery system in therapeutic applications. For example, liposomes can be used to introduce contract agents into the body for diagnostic imaging procedure, including computerized tomography CT and magnetic resonance imaging MRI. Fluid mosaic model. In 1972, S.J. Singer and G.L. Nicholson proposed the fluid mosaic model for membrane structure, which suggested that the membrane or dynamic structure composed of proteins and phospholipids. In this model, the phospholipid bilayer is a fluid matrix, a two-dimensional solvent for proteins. Both lipids and proteins are capable of rational and lateral movement. Singer and Nicholson also pointed out that the proteins can be associated with the surface and this bilayer or embedded in the bilayer to vary degrees. They define two classes of membrane proteins. The first, called peripheral proteins or extrinsic proteins, includes those that do not penetrate the bilayer to any significant degree and are associated with the membrane by feature of ionic interaction and hydrogen bond between the membrane surface and the surface of the protein. The peripheral proteins can be dissociated from the membrane by the treatment with salt solution or by changes in the pH. Treatments that disrupts the hydrogen bond and ionic interaction. Integral proteins or intrinsic proteins, in contrast, possesses the hydrophobic surfaces that are readily penetrates the lipid bilayer itself, as well as the surfaces that prefers contact with the aqueous medium. These proteins can also insert into the membrane or extend all the way across the membrane with the aqueous medium. The proteins can also insert into the membrane or extended all the ways across the membrane with the aqueous medium. These proteins can also insert into the membrane or extends all the way across the membrane and expose themselves to the aqueous solution on both sides. Singer and Nicholson also suggests that the portion of the bilayer lipid interacts in a specific way with the integral membrane proteins, and that these interactions might be important for the function of certain membrane proteins. Because of this intimate association with the membrane lipid, the integral protein can only be removed from the membrane by agent capable of breaking up the hydrophobic interactions within the lipid bilayer itself, such as detergents and organic solvents. The fluid mosaic model has become the example for modern study of membrane structure and functions. Membrane bilayer thickness. The Singer and Nicholson model suggests a value of approximately 5 nanometer for membrane thickness, the same thickness as the lipid bilayer itself. Hydrocarbon chain orientation in the bilayer. An important aspect of membrane structure is the orientation or ordering of lipid molecules in the bilayer. Membrane bilayer molecules. The idea that the lipid and protein could move rapidly in biological membrane was a relatively new one when the fluid mosaic model was proposed. 1.2 Structure of membrane protein. The lipid bilayer constitutes the fundamental structure unit of all biological membrane. The protein, in contrast, carry out essentially all the active functioning of membranes, including transport activities, receptor functioning, and other related processes. As suggested by Singer and Nicholson, most membrane proteins can be classified as peripheral or integral. The peripheral proteins or globular proteins that interacts with the membrane mainly through electrostatic and hydrogen bonding interactions with the integral protein. Although the peripheral proteins are not discussed further, many proteins of this classes are described in context of other discussion throughout this book. The integral proteins are those that are strongly associated with the lipid bilayer, with a portion of protein embedded in or extended all the way across the bilayer. Another classes of protein not anticipated by Singer and Nicholson. The lipid anchored proteins are important in a variety of functions in different cells and tissues. These proteins are associated with the membrane by means of varieties of covalently linked lipid anchor. Integral membrane protein. In the face of diversity of integral membrane protein, most fall into two general classes. One of these includes the protein attached or anchored to the membrane by only a small hydrophobic segment, such that most of the protein extends out into the water solvent on one or both sides of the membrane. A protein with a single transmembrane segment. In the case of protein that are anchored by a small hydrophobic polypeptide segment, that segment often takes the form of single helix. One of the best examples of membrane proteins with such an alpha helix structure is glycophorin. Most glycophorin mosses is oriented on the outside surface of the cell, exposed to the aqueous miliae permanently of the plasma membrane. The plasma membrane is differentially selectively permeable. Some substances can move across the membrane and some cannot. The macromolecules cannot diffuse across the membrane because they are too large. The ions and charged molecules cannot cross the membrane because they are unable to enter the hydrophobic phase of the lipid bilayer. The non-charged molecules such as alcohol and oxygen are lipid soluble and therefore can cross the membrane with ease. They can slip between the hydrophilic heads of the phospholipids and passes through the hydrophobic tails of the membrane. Small polar molecules such as carbon dioxide and water also have no difficulty crossing through the membrane. The membrane transport mechanism and ion pump. The transport process are vitally important to all life forms because all cells must exchange the materials with their environment. The cells must obviously have way to bring nutrients into the cell and ways to send waste products and toxic substances out. Also, inorganic electrolytes must be able to pass in and out of the cell and across organelle membrane. All cells maintains concentration gradients of various metabolites across their plasma membrane and across the cell of intracellular organelles. By their very nature, the cell maintains a very large amount of potential energy in the form of such a concentration gradient. Sodium and potassium ion gradient across the plasma membrane mediates the transmission of a nerve impulse and the normal function of the brain, heart, kidney, and liver, among other organs. The storage and release of calcium from cellular compartments controls muscle contraction and the response of many cells of hormonal signals. The high acid concentration in stomach are required for the digestion of food. Extremely high hydrogen ion gradients are maintained across the plasma membrane or mucosal cell lining the stomach in order to maintain high acid level in the stomach wall from the harmless effect of such acid. Some transport protein only provided a path for the transport species, whereas other couples and enzymatic reactions with the transport event. In all cases, the transport behavior depends on the interaction of transport proteins not only with the solvent water but with the lipid milieu of the membrane as well. Figure: passing of molecules into and out of the cell. Diffusion and osmosis. Diffusion is the movement of molecules from a higher to lower concentration, that is, down their concentration gradient until equilibrium is achieved and they are distributed equally. Diffusion is a physical process that can be observed with any type of molecules. For example, when a crystal of dye is placed in water, the dye and the water molecules move in various directions, but their net movement which is the sum of their motions, is towards the region of lower concentration. Therefore, the dye is finally dissolved in the water, resulting in the colored solution. A solution contains both a solute, usually a solid, and the solvent, usually a liquid. In this case, the solute is the dye and the solvent or the water molecules. Once the solute and the solvent are eventually distributed, they continues to move about, but there is no net movement of either one in any direction. As discussed, the chemical and physical property of plasma membrane allows only a few types of molecules to enter and exit the cell simply by diffusion. The gases can diffuse through the lipid bilayer. This is the mechanism by which the oxygen enters the cell and carbon dioxide exits cells. Also consider the movement of oxygen by the alveoli, air sacs of lungs to blood in the lungs capillary. After inhalation, breathing in, the concentration of the oxygen in the alveoli is higher than in the blood. Therefore, oxygen diffuses into the blood. Figure process of diffusion. Diffusion is a spontaneous and no chemical energy is required to bring it about. A. When dye crystals are placed in water, they are concentrated in one area. B. Dye dissolves in water, and there is a net movement of dye molecules with higher to lower concentration. There is also a net movement of water molecules from higher to lower concentration. C. Eventually, the water and the dye molecules are equally distributed throughout the container. Osmosis solutions. Osmosis. The osmosis is a diffusion of water across a differentially permeable membrane from its higher concentration to a lower concentration. The osmotic pressure is hydrostatic pressure, one side of the membrane with higher solute concentration produced by the water diffusing to that side of the membrane. The tonicity is the strength of the solution in relationship to osmosis determines the movement of water into or out of the cells. 1. Isotonic is the relative solute concentration of two solution equal. 2. Hypotonic is a relative concentration of one solution is less than other solution. 3. Hypertonic is relative solution concentration of one solution is greater than another solutions. Active transport. During the active transport, the ions or molecules moves through the plasma membrane, accumulating also inside or outside the cell. For example, iodine collects in the cell of thyroid gland. The nutrients are completely absorbed from the gut by the cells lining the digestive tract, and sodium ions Na plus can be almost completely withdrawn from urine by cells lining the kidney tubules. In these instances, the substances have moved to the region of higher concentration, exactly opposite to the process of diffusion. It has been estimated that up to 40% of cells' energy supply may be used for active transport of solute across the membrane. Both carrier protein and an expenditure of energy are needed to transport the molecules against the concentration gradient. In this case, the energy ATP molecules is required for the carrier to combine with the substances to be transported. Therefore, it is not surprising that the cell is involved primarily in the active transport, such as kidney cells have many mitochondria near the membrane through which active transport is occurring. Figure facilitated transport outside and inside. The active transport allows a solute to cross the membrane from the lower solute concentration to higher solute concentration. 1. Molecules enter carrier. 2. Chemical energy of ATP is needed to transport the molecules which exist inside the cells. 3. Carrier returns to its former state. The protein involved in the active transport often are called pumps because as a water pumps uses energy to move water against the force of gravity, the protein uses energy to move a substance against its concentration gradient. One type of pump that is active in all cells but is especially associated with the nerves and muscle cells moves the sodium ion Na plus to to the outside of the cell and potassium ion K plus to the inside of the cells. These two events are presumed by to be linked, and the carrier protein is called the sodium potassium pump. A change in carrier shape after the attachment and again after the detachment of phosphate group allows the carrier to combine alternatively with the sodium ions and potassium ions. The phosphate group is donated by ATP, which is broken down enzymatically by the carrier. The passage of salt NaCl across the plasma membrane is of primary importance in the cell. The chloride ion Cl usually crosses the plasma membrane because it is attracted by positively charged sodium ions. Na plus. The first sodium ions are pumped across the membrane, and then chlorine ion simply diffuses through the channel that allows their passage. As noted in above, the chloride ion channel many functions in persons with cystic fibrosis, and this leads to the symptoms of this inherited genetic disorder. A carrier protein actively moves three sodium ions Na plus to the outside of the cell for every potassium ions K plus pumped inside the cells. Note that the chemical energy of ATP is required. Inactive transport molecules are transported away from the thermodynamic equilibrium. Hence, energy is required. This energy comes from the hydrolysis of ATP, from the electron movement, or from the light. The maintenance of electrochemical energy comes from the hydrolysis of ATP, from the electron movement, or from the light. The maintenance of electrochemical gradient in biological system is so important that it consumes about 30 to 40% of total energy expenditure in a cell. In general, the cell maintains a low intracellular Na plus concentration and a high intracellular K plus concentration, all along with the net negative electrical potential inside. The pump that maintains these ingredients is an ATPase that is activated by Na plus and K plus. The ATPase is an integral membrane protein and requires phospholipids for activity. The ATPase has catalytic center for both ATP and Na plus on the cytoplasmic side of the membrane, but the K plus binding site is located on the extracellular side of the membrane. Aubane or digitalis inhibits this ATPase by binding to the extracellular domain. The inhibition of ATPase by Aubane can be antagonized by extracellular K plus. Figure the K plus the Na plus K plus ATPase pump. This pump moves three Na plus ions from outside of the cells to the outside and brings two K plus ions from the outside to the inside for every molecules of ATP hydrolyzed to the ADP by membrane associated ATPase. Passive transport means name, diffusion, direction towards lower concentration. Requirement concentration gradient only. Examples: lipid soluble molecules, water and gases. Passive transport means name, facilitated transport, direction towards lower concentration. Requirement carrier and concentration gradient. Examples: some sugar and amino acids. Active transport means name, active transport and direction towards great concentration. Requirement carrier plus cellular energy. Examples: other sugar, amino acids and ions. Exocytosis directions towards outside. Requirement vesicle fuses with plasma membrane. Example: macro molecules. Endocytosis, phagocytosis. Name, towards inside requirement vacuoles formations, examples cells and subcellular materials. Active transport means by name. The pinocytosis includes receptor mediated endocytosis. Direction towards inside requirement physical formations and examples on the macromolecules. Ion channels. Ion channel provides the hydrophilic pores through which selected ions can diffuse, move down their electrical or chemical concentration gradient. They are characteristically unsaturable and have very high flux rate. Many ion channels are highly specific for one ion, and most are gated by either voltage or a ligand. In bacterial, the K plus channels or selectivity filter provides ligands or voltage gated. The acetylcholine receptor channel is gated by the uh, acetylcholine, which triggers subtle conformational changes that opens and close the transmembrane path. Ion channel together with within pumps such as the sodium Na plus, potash K plus, ATPase determines a plasma membrane permeability to specific ion and regulates the cytosolic concentration of ions and the membrane potential. The properties characterize ion channels. 1. Ion channels can be highly selective for ions. 2. Ion channels exist in open and closed state. 3. Most cells have a varieties of sodium Na plus, K plus, Ca2 plus, Cl chloride channels. 4. The open state of channels often spontaneously converts into inactivated conformations. 5. The transition between the open and closed state are regulated. The ion channels are divided into two classes, the ligand gated channels and voltage gated channels. The ligand gated channels opens and close in response to the binding of specific chemicals, whereas the voltage gated channels opens and close in response to the electrical potential across the membrane. The most organisms contains many ion channels. One of these, which plays a role in the nerve conditions, is the voltage gated K plus channel. It is closed most of the time, but open when a nerve impulse arrives, dropping the membrane potential from its resting 50 to 70 millivolt negative inside value to below zero. This voltage channel opens the channel this voltage change opens the channel, allowing a very rapid outflow of K plus ions. The channel then closes spontaneously. This conduction channel is lined largely with the hydrophobic group. Figure: ion channel open in response to a special stimulus. Ionophores. Ionophores, certain microbes synthesis is small organic molecules, ionophores, that functions as a shuttle for the movement of ion across the membrane. These ionophores contains a hydrophilic centers that binds specific ions and are surrounded by a peripheral hydrophobic regions. This arrangements allows the molecules to dissolve effectively in the membrane and diffuse transversely therein. Examples: polypeptide gramicide form channels, aquaporins. In certain cells, example red cells of collecting ductals of kidney, the movement of water by a simple diffusion is increased by movement through water channel. These channels are composed of a tetrametric transmembrane protein and named aquaporins, a family of integral protein discovered by Peter Agri. The aquaporins AQPs provides a channel for rapid movement of water molecules across all plasma membrane. These are distinguished from ion transporters in at least three ways. First, the rate of flux through channels or by several orders of magnitude greater than the turnover number for transporter. Second, the ion channel are not saturated. The rate do not approaches a maximum at a high saturated concentration. Third, they are gated, open or closed in response to some cellular events. One, ligand gated channel, generally oligomeric. A binding of an extracellular or intracellular small molecule forces an allosteric transition in the protein which opens or closes the channel. 2. Voltage gated ion channels. A change in transmembrane electric potential Vm causes a charged protein domain to move relative to the membrane, opening or closing the ion channel. The both type of gating can be very fast. A channel typically opens in a fraction of a millisecond may remains open for only milliseconds, making these molecular devices effective for very fast signal transmission in the nervous system. Some disease resulting from ion channel defects. Ion channel disease. Ion channel Na plus voltage gated and skeletal muscle disease, hyperkalemic periodic paralysis or myotonia congenita. 2. Na plus voltage gated and neuronal disease, generalized epilepsy with febrile seizure. Ion channel Na plus voltage gated and cardiac muscles disease, long QT syndrome. 3. Ion channels Ca2 plus neuronal disease, familial hemiplegic migraine. Ion channel Ca2 plus voltage gated and retina disease, congenital stationary night blindness. Ion channel Ca2 plus polycystin 1 disease, polycystic kidney disease. Ion channel K plus neuronal dominant deafness. Ion channel K plus voltage gated and neuronal benign familial neonatal convulsion. Ion channel non-specification CGMP gated and retinal disease, retinitis pigmentosa. Ion channel acetylcholine receptor skeletal muscle disease, congenital myasthenic syndrome. Ion channel Cl minus cystic fibrosis disease. Ion pump. The cell have an active ion pump that removes Na plus from the cell and introduces Ka. For example, the cytoplasm of the giant axons of the nerve of squid can be squeezed out and replaced by the ionic solution. And Na plus K plus activated ATPase, often abbreviated Na plus K plus ATPase, as synonymous with the membrane-bound ion pump. Within the cell, Na plus is located on one side of the membrane and K plus on the other to activate this enzyme. The sodium potassium pump displays a curious stoichiometry. 3 sodium ion are pumped from the inside and 2 potassium ion from the outside of the cell for each molecules of ATP cleaved. Thus, an excess of positive ion is pumped out with the results that a negative charge developed inside the cell and a positive charge accumulates on the outside. This action of the Na plus K plus ATPase is the primary source of membrane potential for most eukaryotic cells and is said to be electrogenic. The P-type ATPase are characterized by phosphoenzyme intermediates and through inhibition by vanadate ion. In addition to the Na plus K plus ATPase, there is a very active Ca2 plus ATPase which transports two Ca2 plus from the inside of the cell to outside while returning 2H plus from outside per ATP. 1.3 Mechanism of sorting and regulation of intracellular transport. The protein biosynthetic pathway in cell can be one large sorting system. Many proteins carry signals usually but not always specific sequence of amino acids that directs them to their destination, thus ensuring that they will end up in the suitable membrane or cell compartment. These signals are a fundamental components of the sorting system. Usually, the signal sequence are recognized and interacts with the complementary area of the protein that serves as a receptor for the protein that contain them. The major sorting decision is made early in the protein biosynthesis when a specific portion are synthesized either free or on membrane bound polyribosomes. These results in two sorting branches called the cytosolic branches and the rough endoplasmic reticulum or branch. This sorting occurs because the protein synthesized on the membrane bound polyribosomes contain a signal peptide that mediates their attachment to the membrane of the endoplasmic reticulum. Further details on the signal peptide are given below. The protein synthesized on free polyribosome deficiency, this signal peptide and are derived into the cytosol. These are directed to chloroplast, mitochondria, nuclei, and peroxisomes, but by a specific signal or remain in the cytosol if they lack a signal. Any protein that contains a target sequence that is subsequently removed is designated as a pre-protein. Figure. The diagrammatic representation of two branch of protein sorting occurred by a synthesis on cytosolic and two membrane-bound polyribosomes. The mitochondrial protein listed are encoded by a nuclear gene. Some of these signals used in further sorting of this protein are listed in table. Polyribosomes 1 cytosolic, 2 rough endoplasmic reticulum. Cytosolic proteins mitochondria, nuclear, peroxisomal, and cytosolic. 2 rough endoplasmic reticulum. Membrane, GA membrane, plasma membrane, secretory, lysosomal enzymes. In some cases, a second peptide is also removed, and in that even the original protein is known as a pre-pro protein. Example, pre-proalbumin. The protein synthesized and sorted in the rough branch in figure includes many destined for various membranes. Example of endoplasmic reticulum, Golgi apparatus, lysosomes, and plasma membrane. And for secretion, the lysosomal enzymes are also included. Thus, such proteins may reside in the membrane or lumen of the endoplasmic reticulum or follow the major transport route of intracellular proteins to the Golgi apparatus. Further signal mediated sorting of certain proteins occurred in the Golgi apparatus, resulting in delivery to lysosomes, membrane of Golgi apparatus, and other sites. The protein destined for the plasma membrane or for secretion passes through the Golgi apparatus, but generally are not through to carry specific sorting signals. They are believed to reach their destinations by default. A. Free ribosomes in systole. B. Membrane-bound ribosomes. The entire pathway of endoplasmic reticulum to Golgi apparatus plasma membrane is often called as a secretory or exocytosis exocytotic pathway. The events along this route will be given special attention. Most of the protein reaching the Golgi apparatus or the plasma membrane are carried in transport vesicles. Diagrammatic representation of a rough endoplasmic reticulum branch of protein sorting. Other protein des- destined for secretion are carried in the secretory vesicle. These are prominent in the pancreas and certain other glands. Their mobilization or discharge are regulated and often referred to as a regulated secretion, whereas the secretory pathway involving transport vesicle is called constitutive. There is a considerable intracellular traffic of protein. The Golgi apparatus plays a major role in the sorting of proteins for their correct destinations. The protein biosynthetic pathway in the cells can be one large sorting system. Many proteins carry signals the sequence of amino acids that directs them to their destination. The entire pathway of endoplasmic reticulum, Golgi apparatus, plasma membrane is often called the secretory or exocytotic pathway. The Golgi apparatus or the plasma membrane are carried in the transport vehicles. A brief description of the formation of these important particles will be given subsequently. Other protein destined for secretion are carried in the secretory vesicles. Proteins that are transported by the vesicles are called cargo. These are prominent in the pancreas and certain other glands. Their mobilization and discharge are regulated and often referred to as regulated secretion, whereas the secretory pathway involving transport vehicle is called constitutive light. The translocation is occurred post-translationally after the matrix protein and are released from the cytosolic polyribosomes. The interaction with a few cytosolic proteins that acts as a chaperones, the protein folding machineries RER, and as targeting factor occurs before translocations. The figure diagrammatic representation of a rough endoplasmic reticulum branch of protein sorting. The signal hypothesis was proposed by Blobel and Sabatini to explain the distinction between free and membrane-bound polyribosomes. They found that the protein synthesized on membrane-bound polyribosomes contained a peptide extension signal peptide at their amino terminus, which mediates their attachment of a membrane of endoplasmic reticulum. The mRNA for secreted protein encoded an amino terminal signal peptide leader sequence. The protein is inserted into the endoplasmic reticulum membrane at the same time as its mRNA is being translated on polyribosomes, so-called co-translational insertion. As this signal peptide emerges from the large subunit of ribosome, it is recognized by a signal recognition particle SRP. The signal recognition particle contains 6 protein and has a 7s RNA associated related to the ALU family of highly repeated DNA sequence. The SRP imposes a block until the SRP signal peptide polyribosome complex has bound to the so-called docking protein SRP or a receptor for the SRP on the ER membrane. The SRP thus guides the signal peptide to the SRPR and prevents premature folding and repulsion of the protein being synthesized into the cytosol. Diagram. The diagram of single hypothesis of transport of secreted protein across the ER membrane. The insertion of single peptide into the conducting channel while the other end of the parent protein is still attached to the ribosome is termed co-translational insertion. The routes of protein assembly. The protein follows several routes to be inserted into or attached to the membrane of the endoplasmic reticulum. A. 1. Co-translational insertion. 2. Synthesis on free polyribosomes and subsequent attachment of endoplasmic reticulum, and retention of luminal aspects of the endoplasmic reticulum by a specific amino acid sequence. 4. Retrograde transport from the Golgi apparatus. The figure, the flow of membrane protein from the endoplasmic reticulum to cell surface. The horizontal arrows denotes steps that have been proposed to the signal independent and thus represents bulk flow. To cis Golgi, to medial Golgi, and to trans Golgi to lysosomes and to cell surface, and trans Golgi to secretory storage vesicles. Some sequence that direct protein to a specific organelles. Targeting sequence. Targeted signal peptide sequence. Organelle targeted membrane of endoplasmic reticulum. Targeting sequence. Amino terminal KDEL sequence. Lysine, aspartame, glycine, leucine. Organelle targeted luminal surface of endoplasmic reticulum. Targeting sequence. Amino terminal sequence 20 to 80 residue. Organelle targeted mitochondrial matrix. Targeting sequence NLS, nuclear location signal. Example: proline 2, lysine 2, alanine, lysine, valine. Organelle targeted nucleus. PTS, peroxisomal matrix targeting sequence. Example: serine, lysine, leucine, peroxisome. Targeting sequence mannose 6 phosphate. Organelle targeted lysosome. Chaperone protein. Many are so-called heat shock protein HSP. Some are inducible by conditions that causes unfolding of newly synthesized protein. They bind to predominantly hydrophobic regions of unfolded and aggregated proteins. Most chaperones show associated ATPase activity with ATP or ADP being involved in the protein chaperones interactions found in various cellular components such as cytosol, mitochondria, and the lumen of endoplasmic reticulum. Coated vesicles. The vesicles are used to transport proteins both out of the cell and into the cell. The secretion of protein is called exocytosis. Internalization of a protein is called endocytosis. The protein are transported in coated vesicles. The constitutive bulk flow transport from endoplasmic reticulum through the Golgi takes place by COP coated vesicles. The Catherine coated vesicles are used for both regulated exocytosis and endocytosis. Key points: the protein transport through the endoplasmic reticulum Golgi system occurs in the coated vesicles. 2. The secreted proteins moved in the forward anterograde directions towards the plasma membrane. 3. There is no movement within the system in the reverse retrograde directions. 4. The proteins that are improved through the plasma membrane also are incorporated in coated vesicles. 5. The coated vesicle bud from a donor membrane surface and fuse with a target membrane surface. The endocytic and secretory vesicle have a clathrin as the most prominent protein in their coats and are therefore known as a clathrin coat vesicles. The subunit of a coat consists of a triskelion, a triple pronged protein complex. Figure: clathrin coated vesicles. The inner shell of the coat is made by protein called adaptor, which binds to clathrin and to a integral membrane protein of vesicles. The COPI coated vesicles are responsible for retrograde transport from the Golgi to the endoplasmic reticulum. The COP2 vesicles undertake forward movement from the endoplasmic reticulum to Golgi. In the pathway for regulated secretion of protein, the protein are sorted into the catherine coated vesicles as the Golgi transphase. Some vesicles may fuse into large secretory granule. The vesicle also moves to endosomes, which controls trafficking to the cell surface. The secretory vesicles are stimulated to upload their cargoes at the plasma membrane by extracellular signals. Some vesicles are used for endocytosis, the pathway by which the protein are internalized from the cell surface. 1.4 Electrical properties of membrane. All cells are surrounded by the cell membrane. It is the simplest form. It is a phospholipid layer, that is a layer which is only two phospholipid molecule thick. Due to the amphipathic nature of lipid, artificial cells can be made from its constituent phospholipid molecules by suspending in water and are termed as liposomes. We will neglect all the complexities of the metabolic and structural apparatus found in the interior of the cell and simply considered it as a little bag formed by the cell membrane and filled with the saline, that is water with ion dissolved in it. Equally, we will assume that the exterior of the cell is a bath of saline. 1. Conductance. The artificial membrane, we know that pure phospholipid bilayer are quite good insulator. That is not surprising. There are no free ion in the membrane, so there are no carrier to transport charges. Their specific conductance per unit area is only about G pure is equal to 10 to the power of minus 13 delta minus 1 m minus 2. The conductance of a biological membrane is much higher, typically by several order of magnitude, even at rest, that is without synaptic influence. 2. Capacitance. According to our simplification, the inside and outside of the cell are both solutions of various salt in water. As opposed to the cell membrane, the salt water constitutes quite a good conductor because there are free ions that are transport electrical charges. 3. Electrical potential across the membrane. Our interest is mainly on functions of neuron, which is a class of cells that uses electrical signals for information processing. How can a cell generate such signals? The first thing we need is some way of generating a different voltage at different parts of the system inside and outside of the each cell. We know that the relationship between these probabilities is given by the Boltzmann distribution. P in by P out is equal to x minus of E in by KT divided by x of E out minus of E out by KT, where K is constant called as a Boltzmann factor and T is a temperature in Kelvin. The detail are not important for our purpose. We can rewrite about the equation in form P in by P out is equal to x exp minus E in minus E out divided by KT. Therefore, all our energy terms calculated out, and we will left with P in by P out is equal to EXP minus ZE of V in minus V out by KT. Now we can turn things around. Instead of computing the probabilities from the voltage, the voltage difference between the inside and the outside of the cell is obtained as delta V is equal to V in minus V out is equal to KT by ZE in P out by P in. The formation or equivalent. It is our customary to set the voltage scale such that the V out is equal to 0. We thus find that each ion species has its own voltage at which it is in statistical equilibrium. This voltage is commonly called the reversal potential of this ion because the current generated by this ion reverses its sign when this voltage is applied to the membrane.